Good evening, everyone. Welcome back to my channel. After a long time, I'm going to continue with the topic where I left. I started with sexual reproduction in plants where I completed up to endosperm. So now I would like to continue with that and let us. So after that, we have to continue with the development of dicot embryo. So let us continue with that. So first, what is embryogenesis? First, we have done about endosperm, how endosperm is formed, and there is a need of endosperm formation much before the embryo development because to ensure proper nutrition to the embryo. This is a basic principle of nature that before actually a life is developed, so nutrition has to be provided to that particular life. So therefore, before actually embryo grows and develops, so it is a necessity that the endosperm must first develop. So prior to embryo development, prior to embryogenesis, there will be development of endosperm. After some amount of endosperm is developed, then the embryo development takes place. Now, where does this embryo development takes place? Where exactly the embryo is formed? Embryo is formed in the place where there is a position of or where there is actual position of egg cell. In an ovule, we know the egg cell is placed towards the micropylar end. So therefore, since process of fertilization occurs at that micropylar end, so there will be zygote formation and then consequently an embryo will be formed. So the basic development of embryo in both dicots and monocots is almost the same but with few differences. In today's session, I am going to discuss about embryo development in dicots. So let us see. So here I have already illustrated with some diagrams. So let us try to elaborate them and let us try to understand. And then I will give you the structure of dicot embryo. First let us see the embryo development, how embryo starts developing. So we know all that the first cell of embryo is the zygote. Zygote always will be the first cell and this zygote will initially undergo one mitotic division. This is one transverse division a zygote will undergo. And as a result of this transverse division, it will develop two cells, embryonal cell and suspensor cell. This embryonal cell, it is also named as, it is a terminal cell. It is also called as terminal cell and the suspensor cell is named as a basal cell. So there is a terminal cell and there is a basal cell. Now further, so first division according to this, what is the first division? It is a transverse division. Now the second division, let us see how it takes place. Now let us see the second division, how it takes place. So second division, during second division, this I told you the upper cell, it is a suspensor cell or basal cell, lower cell is the terminal cell. So this upper cell or suspensor cell will undergo one transverse division. So this division, one transverse division, remember the upper or the basal cell will undergo one transverse division and the lower cell or we call it as terminal cell, this will undergo one longitudinal division. So after one transverse and one longitudinal division, we find this is a T-shaped embryo is formed. This T-shaped embryo is called as proembryo. This T-shaped embryo is called as a proembryo. Now, in the next stage, this is also called, this now further only this lower part, that is this embryonal proper. This is called as embryo proper. So this terminal cell which has divided to form two cells, this will undergo further development. So this will undergo another transverse division. It will undergo another transverse division to form to form 4 cells. To form 4 cells. That's what we have shown in this diagram. To form 4 cells. And what is this stage of pro-embryo called? This is called as a tetrad stage. Second one is called as a dyad stage, 2-celled stage. Next one, this is called as a tetrad stage. Now, after the tetrad stage, further divisions will occur all transversely. So, each cell will undergo a transverse division. And after it undergoes a transverse division, next we will see an 8-celled embryo or an octant is formed. 8-celled embryo is called as octant. So, an octant is formed. Now further, these cells will divide and then they get differentiated into these 3 layers will be formed. These 3 layers are the embryonal layers which are formed. And this is called as, so what are these different stages? First we are starting with zygote, 2-cell stage, 4-cell stage, 8-cell stage. After 8-cell stage, this stage will be called as a globular stage. This stage we call it as a globular. So first stage we are starting as a 2-cell stage, 4-cell stage, 8-cell stage. All these 3 stages come under pro-embryo stage. First is zygote, then pro-embryo, then globular stage, heart-shaped and mature embryo. These are the 5 stages in the development of dicot embryo. So in the globular stage, when it completely develops, there are 3 layers formed. Outer layer of cells form dermatogen, outer layer of cells. So this outer layer of cells which form dermatogen, they should give rise to the outermost layer called as epidermis. It gives rise to the outermost layer called as epidermis. Similarly, the next one, middle layer, which is called as a ground meristem. Middle layer is called ground meristem. Ground meristem. This ground meristem cells, when they differentiate, they give rise to the ground tissue system of the root. They give rise to ground tissue system of the root. Then next, the innermost layer. Innermost layer is called as a procambium. So the cells of procambium, when they differentiate, they give rise to vascular bundles. Cells of procambium, when they divide, they form the vascular bundles. So in this way, we can see the 3 layers. They are called germinal layers. That means they are also embryonal layers. They give rise to the 3 different layers. This is called as a globular stage. Now the next stage is called as a heart-shaped stage. So now let us see. Now let us go to the next stage. We have done up to where? We have done up to the globular stage where the 3 layers are formed and then they get differentiated to form the different types of parts of the different tissues. Now next stage, it gets differentiated to form heart-shaped. Meanwhile, when it is forming the heart-shaped, we have the upper cell, that is suspensor cell. Suspensor cell, or we call it as a basal cell. This will undergo further division and it will form about 6 to 10 cells. So basal cell divides to form 6 to 10 cells in a single row. They form the cells in a single row. Out of these, only one cell, one which is at the top, one cell will differentiate to form the tip, that is called as this cell is called as haustorium. It is called as haustorium. The tip, the topmost cell. Remaining about 10 cells, they form the suspensor. Now what is the function of this haustorial cell and suspensor cell? Haustorial cell will help in absorption of food The embryo needs food, and suspensor will push the embryo deep into the endosperm. So I am going to write here. The basal cell divides to form 6 to 8 cells. One of the cell develops into haustorium. One of the cell develops into haustorium, which help in absorption of food material. From where it has to absorb food material? From the surrounding endosperm. Endosperm is already formed, and this embryo is placed in the endosperm. So therefore, it will absorb food material from the endosperm. Now, what is the use of the suspensor? Let us see. So, suspensor helps in. So, remaining cells will form suspensor. Remaining cells form suspensor, and the function of suspensor is it pushes the embryo into the endosperm. So now we have seen how it develops. Starting from single cell, first two cells are formed, upper and lower or basal and terminal. Terminal cell is called as embryo proper, which again further divides and forms the embryo. Globally, it reaches, after reaching up to globular embryo, then it forms the 3 embryonal layers. Each embryonal layer, depending on its location, it gives rise to one particular part of the root or one particular tissue of the root. Then the basal cell will further divide, and it forms 6 to 10 cells. Out of that, one cell will develop into haustorium, which helps in absorption of food material from the surrounding endosperm. The remaining cells will form suspensor, and that suspensor will help in pushing the embryo deep into the endosperm. After this stage, globular stage, the embryo now assumes a heart shape, and then finally the embryo becomes completely mature. This is called as a mature embryo. So a mature embryo will have suspensor and haustorium are reduced, and a radical is developed, plumule is developed, and these are the two cotyledons also will develop. This is how the dicot embryo develops. Now I will give you briefly about what is the structure of dicot embryo. So let us see the structure of dicot embryo. So when we look at the dicot embryo, a dicot embryo will have an axis. This is called as embryonal axis. It is called as embryonal axis. This embryonal axis, at the tip of embryonal axis, this is called as a plumule, and at the base it is called as a radical. And this embryonal axis is also called as tigellum. Embryonal axis is also called as tigellum. It is also called as a tigellum. Now the just below the plumule, and there will be two cotyledons present. So a region present just below the plumule and above the cotyledon. This region is referred as epicotyl. Similarly, the region which is present below the cotyledons, it is called as hypocotyl. And here radical is also covered by a root cap. Some of the cells at this stage, especially which are forming dermatogen, some cells of dermatogen, they also give rise to root cap. We can find here the root cap. These are the some cells of dermatogen, or we can say basal cell of suspensor, the last cell of suspensor that can give rise to a root cap. So the root tip will be covered by a root cap. The axis we are calling it as a tigilla. Externally, if we take a dicot seed, a dicot seed will have a seed coat, outer testa, inner tegment. Two distinct seed coats are present, which are not found in monocots. So distinct two seed coats will be present. And the seed will have a scar, a clear scar, like if we observe a pea seed, a pea seed will have a scar. This scar indicates the point of attachment of seed and the fruit, and this is called as a micropyle, which is the seed pore. Seed pore helps in absorption of water during seed germination. So I think with that we conclude about dicot embryo and about the dicot seed. So this session I will conclude my video. In my next video I am going to describe about development of monocot embryo. That's all for now. All the best to all my students. Please do listen, subscribe and share. Thank you so much.